Could you say a little bit more about this idea of neurodiversity and how it relates to Neanderthals?
Well, we have in our own population a variety of individuals who cover a spectrum of neurodiversity, meaning the high-functioning autistics, dyslexics, narcoleptics, even bipolar have been major world leaders and thought leaders, despite or possibly because of their neurodiversity.
For the first time ever, researchers have presented the gene sequence of an extinct prehistoric human. The geneticist Svante Pääbo, one of the directors of the Institute, has been working on decoding the Neanderthal genome since 1997.
If we want to pinpoint what is unique in terms of genetics about the human being today, then we can compare ourselves not just with chimpanzees, with whom we share a common ancestor of between 5 to 7 million years ago. We can also compare ourselves with the relative who's closest to all of us, the Neanderthal, with whom we share a common ancestor of 300 to 400,000 years ago. We can say which mutation in the genome is unique. This is the first really important objective. We can then compile a catalogue where we have the strongest evidence of positive selection. What I find extremely interesting is that there are several areas related to mental development, for example, mutations which lead to autism or schizophrenia in people today. This suggests that genes which have something to do with this kind of mental ability mutated. Of course, this doesn't mean that Neanderthals had this problem which led to this mutation. But it does show that these genes in this area somehow changed.
Because we coexisted for a long time, there was this open question forever whether there's any genetic relatedness between humans and Neanderthals. We know we lived at the same time and overlapped geographically even. So to address this, there was this analysis where we took the genomes from 5 humans that we sequenced who were from around the world, one person from France, one person from China, someone from Papua New Guinea, someone from South Africa, and someone from West Africa, and took all pairwise combinations, all 2 combinations of each of these, the Yorubas from West Africa and the French guy, and find places where they differ, where there's some allelic difference between these 2 people, and then ask, what does the Neanderthal have? Does it match the person from France or the person from Africa? If Neanderthals are a clean outgroup to all humans, if Neanderthals split off from humans before there was any Yoruban or French or Papua New Guinea, and if all of that was later, then Neanderthals should match these 2 equally often. It should be 50-50. But it turns out, so here are the 5 humans that we sequenced, it turns out that it's not 50-50. Any combination between African and non-African, there was a small but very statistically significant excess matching between Neanderthal and the non-African. There's a closer genetic relationship between Neanderthals and all non-Africans than Africans. And this is interesting because this is the case even for the Papuan sample, even someone from Papua New Guinea, where Neanderthals are never known to have been and almost certainly never were. We still see this signal of admixture in people who live there today from Neanderthals.
As we go further and further afield from our species, we will find we might find additional highly optimal. These are not like these are mutants. These are highly optimal sibling or cousin variations on the human existence.
And so what we understand that in attributing this to an enhanced perceptual expertise. Okay. So this does not precise at which level faces are processed or objects are processed.
What's the applicability of your study?
As the applicability is to consider the autistics as perceptual experts instead of being always lacking of something.
So they're perceptual experts?
They are perceptual experts.
And what is it about the condition of autism that makes it sort of useful to you as a scientist, as a lab head?
So at the beginning, we worked on shape perception. We found a lot of strengths. By strengths, we mean that when we compare autistics to matched groups of people with same age, intelligence, and so on, we see that autistics are not only intact in a certain number of cognitive operations, but they happen to be superior.
Some of the ways in which autistics are superior to normal people would be useful for science, presumably.
Yes. Of course, it needs a combination of several things. For example, general intelligence and to be able to use and do something with his cognitive strengths. But what seems to be a kind of rule is that autistics are able to extract regularities from a larger amount of data than non-autistics. So what we non-autistics do, for example, when we learn language, when we are able to extract what is similar in various sentences to be able to crack the code and to learn words and so on. Curiously, autistics have troubles to do that with, for example, oral speech, but in the visual domain, they are able to extract very complex regularities, and sometimes once they have cracked the code of written material, they are able to detect even mathematical regularities that for us are nearly impossible to remember.
A striking example of this is a young autistic student named Omar, shown here with his dad. Omar had such difficulty with language that he only spoke in single words. Never in full sentences, not even to his parents. And when Omar's school started using our program, his teachers were astonished at how gifted Omar was in math. They never knew. But more amazingly, Omar started talking. And the first full sentences he ever spoke were about these mathematical ideas he was experiencing in these visual games. And he's now excelling in school in both math and English, and it has completely changed his life.
You have a close collaborator called Michelle Dawson, who herself has autism. How did she come to work with you in your lab?
We met her on a clinical basis first, and then I happened to know that she had learned by her own a very complex set of working rules for people with handicap and things like that in a few months, that she was able to dominate a field that would have been a nightmare for me, and that she did that entirely on her own, I think, in libraries and so on. And so I realized that she was exceptional in terms of learning ability. And then I gave her, because as you can hear, I'm not a native English speaker, so I gave her our papers to edit. And she edited the paper in scientific English, which was already something amazing, but also she gave comments on the paper because she had read the entire bibliography of the papers. And specifically, I noticed that she is an inconsistency detector. So even if we thought we had done very, very good work, she was able to say, oh, but you don't say exactly the things the same way with the same understatement in this paper and that paper. And gradually she kind of corrected sometimes minor errors, but sometimes reasoning inconsistencies.
You know, Michelle is somewhat of an exceptional case. Not all autistic people would have skills like this. I mean, what proportion of people with autism are able to have functional working lives?
Oh, that's a quite important question, and it's entirely depending on what you do with them. In the sense that if you look how many autistics have, for example, a normal intelligence, it, as we say in the paper, it entirely depends on the kind of test you use. So the usual number that is in all textbooks is that 3 on 4 autistics are mentally disabled. But when you use nonverbal intelligence testing, this number shrinks considerably. So we cannot know currently the proportion. A large number of autistics can do a secondary and academic studies, even if they are just very intelligent, not exceptionally intelligent. And of course, Michelle is a kind of fairy tale in terms of story of relation of an autistic with science, but we deeply think that it can be applied to a much larger number of autistics.
Neanderthal this is a cast of a Neanderthal. This is lived about 60,000 years ago in France, And as you can see, he has a very large face. Now his face is still fairly vertical, but the face is voluminous. It's huge compared to a human face, much larger. And the face is also stuck forward really far relative to the brain case.
Now, a study by an MU researcher is helping scientists take the next step in discovering the cause. MU autism expert Christina Aldridge found distinct facial differences when comparing typically developing boys to boys who had autism. Aldridge says that this information opens new avenues for researchers to explore. In her study, Aldridge used a 3D imaging system to map 17 points on the face, such as the corner of the eye and the chin. When comparing boys with autism to typically developing boys, she found significant differences in face shape.
On average, they have a broader mouth and philtrum, so the divot above the upper lip seems to be broader in kids with autism, as well as sort of the mid-face region.
One of the areas that I'm particularly interested in is the brain size differences that we see. And we've just finished a study we're actually presenting this morning here at IMPHAR, where we looked at this finding that seems to be now highly replicated, that the brains of children with autism, young children with autism between 1 and 4, go through an accelerated growth, and then typicals catch up. So for a period of time between about 1 and 4 years of age, the brains of children with autism are actually larger.
The first year of a baby's life is one of the most crucial phases of cognitive development, and according to new research at the Max Planck Institute for Evolutionary Anthropology in Germany, the brain development of modern human babies during this time differs significantly from that of our extinct relatives, the Neanderthals. Paleoanthropologist Philip Gunz and his team compared imprints of human and Neanderthal brain cases.
Most people had assumed that all the differences between modern humans and Neanderthals were already established at the time of birth. Both species start off with having a very elongated brain and about the same cranial volume. And then within the first year of life, modern humans get a more globular brain. So the cerebellum expands and the parietal lobe expands and the cranial base flexes. And all of these are things that make modern human skulls unique and also modern human brains unique.
He says without this brief but pivotal period of development, humans would be very different than we are.
We simulated the growth of a modern human baby without this phase of brain development that makes us modern humans so special. And what we found is that they almost looked exactly like Neanderthals. And that's why we think modern humans and Neanderthals did not see the world the same way.
This is endocranial volume means in males on the top and females next. The only disjoint here in this distribution is I don't have a sample of Roman-era females to go in this spot. What happened? Early Upper Paleolithic people who lived 25,000, 15,000 years ago, had bigger brains than Neanderthals. A lot of you who know anthropology have heard Neanderthals had bigger brains than us. It's true. Neanderthals did have bigger brains than us. But they didn't have bigger brains than the people who followed them in Europe. Those people's brains were expanding.
The modern human cranial capacity is smaller than the Cro-Magnon specimen. From this angle, we're seeing something quite interesting. There, this difference in cranial capacity becomes quite obvious. Okay, now this is a comparison of the modern human with a Neanderthal skull, and this Neanderthal skull just goes back and just kind of expands out at the back, which is quite incredible really. It's filling out towards the back. Now this filling out, I think, is quite interesting, because it looks to be the area where the temporal lobe joins to the occipital cortex. In other words, this is the area where the region of the brain which is related to memory is impacting on the occipital cortex, the part of the brain that's related to vision.
Now, I had this brain scan done several years ago, and I used to joke around about having a gigantic internet trunk line going deep into my visual cortex. This is tensor imaging, and my great big internet trunk line is twice as big as the controls. The red lines there are me, and the blue lines are the sex and age-matched control. And there I got a gigantic one, and the control over there, the blue one, has got a really small one. And some of the research now is showing that people on the spectrum actually think with primary visual cortex.
There are some objects which are more, I would say, convincing, like this. Here it's called a pebble. Actually, it's a fossil. It's a small fossil, which is carved with a cross, and this looks really like something intentional and symmetrical. I'm not sure we can prove any kind of aesthetical sense in the Neanderthals with this kind of object, but certainly an attraction for symmetry. And this aspect of being attracted by symmetry is something that has been commented a lot about Neanderthals, mostly because they produced artifacts like this one, which required a lot of skill to be made so symmetrical. And as a matter of fact, we are not quite sure that this would really change very much their efficiency as tools for butchering, for example. But we do have hand axes like this one, which are almost perfectly symmetrical. Experts in flint snapping say that it's quite difficult to produce this kind of object. It's a big investment. It's not easy. And certainly it means something.
But what is particularly interesting about these artworks is not that they're the oldest that we've discovered yet. It's not that we hadn't found them yet in cave systems that we know well. It's the fact that it appears that they were made by Neanderthal as opposed to Homo sapiens. And we didn't think, or at least my understanding here, and let me just double check, we didn't really think that Neanderthal made art. We didn't think that they were sophisticated enough to have made art. And so, yes, we're not sure. But this is also 10,000 years older than the other oldest pieces of art we knew about, which were about 32,000 years old. There were images in the Chauvet cave in southern France. So, yeah, I have no conclusions to draw from it other than... it does say here, as all previous artwork has been attributed to Homo sapiens. So it's going to be interesting to see sort of what conclusions can be drawn out of this. Perhaps there are other pieces of artwork out there as well that were made by the Neanderthal.
On the left is a drawing of a horse done by Nadia when she was 5 years old. She could do this when she was 2.5. She couldn't talk. She had very poor social interactions, but she could draw a horse that would jump off the page. I compare this horse to a horse that Leonardo da Vinci did at the prime of his career, one of the greatest artists of all time. You see, her horse is comparable to his. If you take a normal 8-year-old child, they draw a stick figure of a horse. Now, what is so fantastic is Pablo Picasso pointed out there is no precocity in art. So there are many Mozarts. To be a great composer, many kids start composing at a very early age, 3, 4, 5. Mathematicians can start very early. But Picasso claimed you don't get a sense of perspective, of line and shadow, until you're 9, 10, 11 years old. He could not draw a horse like that when he was 5.
We knew by 2.5 that Dane wasn't talking, but instead he was just expressing himself through art. He was drawing night and day.
Since he was a toddler, Dane has been able to draw with extraordinary precision.
Stephen Wiltshire from London is a star among savants. His nickname is the Living Camera. Stephen is autistic. When he was 11, he drew a perfect aerial view of London after only one helicopter ride. Even the number of windows in all the major buildings in his drawing was correct. After only a 45-minute helicopter flight, we'll ask him to draw a 5.5-yard panoramic picture of the historic city centre. The amazing thing, Stephen starts the drawing as we would, with the Church of St. Peter's. But he doesn't do any sketches. nor roughing out of the space for the drawing. It's as if the panorama already existed within his head. In the left corner, he's finally reached the ruins of the Forum Romanum. Stephen's sister Annette is rejoicing with him. He's made it. Obviously, he's pleased with his work.
One of the things I learned very early on, because I wasn't that social, is I had to sell my work and not myself. And the way I sold livestock jobs is I showed off my drawings, I showed off pictures of things.
And we're back on Radio Freethinker, and we're talking about Neanderthals and all the interesting ideas that are going on right now about our cousins from the past. And it looks like Neanderthals may have beaten humans to the seas. There's growing evidence to suggest that our cousins were accomplished sea travelers. They may have crisscrossed the Mediterranean in boats from 100,000 years ago. Now, not everybody agrees with this theory yet, but there's some interesting evidence that's emerging. We know that they lived around the Mediterranean about 300,000 years ago. Now, we've found their sort of distinctive stone tools on the Greek mainland and, around the Mediterranean, but they've also been found on Greek islands. Now, there's two explanations for this, of course, right? One is that Neanderthals somehow managed to get across the sea to these islands. The other explanation is that maybe these islands weren't islands back then. Maybe there were land bridges or something that was connecting them. So George Friantos of the University of Patras in Greece says we can now rule out the notion that the islands had some kind of land bridge to them. What they figured out was that a long time ago, the sea levels were still very deep, and that they were never shallow enough for a land bridge to go from the Greek mainland to the Greek islands. He suggests that the sea levels were never lower than 120 meters. Ferentinos thinks that Neanderthals created a seafaring culture for tens of thousands of years. Now, to comparison, modern humans are thought to have taken to the seas just 50,000 years ago, and that's how we managed to reach areas like Australia. Now, the journey from the Greek mainland to the Greek islands was not terribly long, maybe 5 to 12 kilometers. But according to some scientists, Neanderthals didn't just stop there. They've apparently found other similar stone tools on islands which are farther away from the mainland, like Crete. And, of course, Crete has been an island for some 5 million years. This, of course, implies that if Neanderthals managed to get to Crete, they probably didn't swim. They probably had some pretty sophisticated boats to get there.
It's a bone with 2 holes that has been described in the literature as a flute, and so people have described this object like the demonstration for the production of music by Neanderthals. Later, it was proved that it's a juvenile femur of bear with 2 holes which have been likely made by the teeth of another bear. And but the fact is that it's possible to produce some kind of music with that, and there are people who have been really good at doing that.
Singing one of my favourite songs from last summer's festivals. This is the Temper Traps, Sweet Disposition.
Now
Derek was excited to show us the skills that make him so exceptional. His ability to instantly call up any piece of music he has ever heard.
Hear a piece once, and you remember it forever.
Yes.
And can you play in almost any style?
I can, yes.
What about YMCA? Can you play that one?
Can play YMCA?
When Derek Paravicini is playing the piano, it's hard to believe there's anything he can't do.
Let's switch to a Russian dance.
So I've been studying what parts of the genome do human populations derive from Neanderthals, and can we make any sense out of what they do? And I will say that this work is really just beginning, and we're learning new things almost every day about it. One of the things that's highly significant about Neanderthal genes in human populations now is that different populations of people outside of Africa, for example, Europeans and Chinese people, have different Neanderthal genes. There's some overlap. There are a few things that are relatively common in Europe and in China and in India and in Japan and in other places. But for the most part, things that are relatively common in Europe are rare in China, and things that are common in China are rare in Europe. It seems that when the mixture took place between humans and Neanderthals, that mixture was before a time that human populations began to separate and become isolated from one another. So the mix that they ended up with is about the same fraction. Look at any person, and he's likely to have about 3% Neanderthal, but the fraction is representing different genes. Most of the genes that came in from Neanderthals are vanishingly rare. Only in 1% of people or fewer. And there's only a few things that are common. So we're beginning to understand the paleobiology that gave rise to our current distribution of Neanderthal genes. Still doesn't tell us what interesting things we might have gotten. But we do know, because we can compare which parts of the genome there are, we do know which genes are involved. So, being a curious anthropologist, I took my own genome and compared it with Neanderthals. It turns out that at a first glance, there are a number of things that I have that came from Neanderthals. It's not yet clear whether the things that I have make any difference to my phenotype, because I'm not looking at the sequence level, and I can't predict when I have a change whether it does something to my anatomical or my behavioral makeup. But I can say that one of the things that I have that has a gene region that's identical to Neanderthals is a part of the androgen receptor. Androgen receptor on the X chromosome is the receptor for testosterone, and that has a huge effect on different elements of the body, both in men and in women. One of the things that it does is it, for some people, increases the likelihood of baldness. And so my version of the androgen receptor, I know, it's likely to correlate with hair loss after age about 25, as it turns out.
Two genetic findings that were published this year, the androgen receptor gene and the sex-related SRCD5, are both associated with autism. Again, for the first time, revealing that there are maybe genes that influence sex steroid hormones, which are atypical in autism.
Now, there's a psychologist at Cambridge University, Simon Baron-Cohen, whom some of you may have heard. He writes of the male mind and the female mind, a politically incorrect statement. But that the male mind is people who like to systematize, who like mathematics, whereas the female mind is people who like social interactions, are empathizing with other people, caring people. So systematizing and caring people. And then you could be put into these two categories, and it's more accurate than you would think. When he looks at the parents of autistic children, that the mothers are more systematizers than on the average, and the fathers are.
There are very clear sex differences in the brain, and we see these differences right from birth. Some people wonder whether these differences in the size of the brain or in the size of different parts of the brain have any impact on the child's psychological development. And certainly we know that girls talk earlier than boys, their language is developing faster, and the regions of the brain that are involved in language are larger in females than in males. The finding that the amygdala is larger in boys is interesting because in boys' and girls' social development, their understanding of other people's emotions, girls seem to develop faster, their empathy is better on average, and this may well relate to this difference in the size of the amygdala. Our research has been looking at whether children and indeed adults with autism might have an extreme of the male brain. So autism is a developmental condition where people have difficulty in developing friendships, relationships, communication, but where they also get very focused, sometimes called obsessional. And we've been interested to see whether the structure of the autistic brain might be an extreme of what we see in the typical male. When you compare groups of children with autism and without autism, children with autism show faster brain development, sometimes thought of as an overgrowth in the early years. And when you actually compare different parts of the brain, some structures fit this extreme male pattern. So the amygdala, which is larger in a typical male, is even larger in a child with autism. The corpus callosum, which is larger in a typical female, is even smaller in a child with autism.
Social functions are localized in the brain just as sensory functions and motor functions are. In fact, we see in the social brain that there are sensory areas and the motor areas. In the sensory areas, the visual system is very important. Faces, as you pointed out, extremely important, reading other people's emotions. So there are face areas in the brain that respond not only to faces, but to emotional expression of faces. There are areas in the brain, sensory areas, that respond to the perception of motion. There's one area that involves response to all motion. Mechanical motion, the movement of a car, as well as the movement of you when you're playing golf. There's another area that this feeds into that is specialized just for social motion. It only responds to biological motion, to you playing golf, to somebody walking, to somebody shaking hands. It does not respond to mechanical motion at all. Moreover, as you pointed out, parts of the motor systems are committed to social behavior. Rizzolatti made this wonderful discovery that there are areas in the motor systems that respond when the monkey picks up a glass of water. That's not surprising. The motor system is designed to pick up a glass of water. But the amazing thing is, those same cells in the monkey respond when you, Charlie Rose, pick up a glass of water. So they are mirroring your behavior. So empathic identification with another person is mediated through the motor system. So we've learned all of this by looking at the emotional brain.
It's been known for some time that children with autism could be quite intelligent, but have a profound deficit in social interaction.
They recorded brainwaves while the kids opened and closed their hands, and while they looked at a movie of somebody else's hands. For most people, the brainwaves look the same either way, whether they're doing or seeing. But for kids with autism, the wave changes, suggesting possibly that autism may have something to do with broken mirror neurons.
Their brains may indeed be different in that regard, and they may have deficits in the mirror neuron system. But we don't know this for sure yet. There needs to be additional work needs to be done using brain imaging.
And one view is that, in fact, in our early ancestry, our hominid ancestry, males and females occupied very different niches, different roles, with females taking much more of a role in childcare and in the community, much more of a social role, and with males having a much more of a solitary role.
There would be very little point in having a mirror system if you lived on your own. There'd be a lot of point in having a digestive system if you lived on your own. There'd be a good point in having a movement system if you lived on your own. There'd be a good point in having a visual system if you lived on your own. But there'd be no point in having a mirror system. The mirror system is the most basic social brain system. It's a brain system which there's no point in having if you don't want to interact or relate to other people.
I have always had the sense that I preferred to spend my time in ways that were not considered socially acceptable. So, you know, I preferred when I was a kid, I liked to go off and do creative projects by myself, and I liked to play with my friends kind of one-on-one. And I was really lucky that I grew up in a family where these things were considered totally normal and great.
I swear I've gone through my life with these various personality traits that I just assumed were like weird little quirks, like feeling a desperate urge to leave a party after an hour, feeling an urge to just be alone in a quiet little place. All these things, and you seem to have spotted that actually this is loads of us. How did you even come across this?
Yeah, you know, that happened, I think, when I was a lawyer. I mean, I became a Wall Street lawyer, of all things, which is a funny career choice, I guess, for someone with this collection of personality traits. And at first I went into my legal career thinking that I was going to be at this big disadvantage because I didn't have the qualities that I associated with being a lawyer. And then I started looking around and realizing that there were a lot of people in my law firm who had the kinds of traits that I did. And that they were using them to good effect, and that I was starting to use them to good effect, and I was trying to understand what this thing was. And at first, the only language that I had for it was gender. So I thought it must be that I'm a woman, and women have these traits of, you know, being able to listen well, and, you know, preparing really thoroughly for meetings, and building good one-on-one alliances behind the scenes, and things like this. But then I started to notice that it was men too. Some men had these traits, and some women did not at all. And that's when I realized it was actually introversion and extroversion, and that there was no real language for talking about questions of identity in the workplace and in society in general. You know, like gender was the only one we had.
So the sort of nasty conclusion from this is that too much mathematical ability is bad for your children. So, you know, I know everyone laughs, but when you get down to it, it's nasty, okay? Because, you know, there's sort of rumors, you know, that more autistic children are born to MIT graduates. There's more autism here in Silicon Valley, okay? Probably true. Simon Baron-Cohen has pretty good data that it's the fetal testosterone, a certain high level of testosterone in sort of the 3rd month of development. And he hasn't shown that that leads to autism, but he's been following, he takes amniotic fluid and then he follows the women afterwards and sees that those which had high testosterone, and then when he does the measurements, that basically it leads to the male mind. Okay. So really what we're trying to do is that autism, because the brain is so complicated, and what we've shown is due to a lot of changes in a lot of different genes, not just one. But there's something strange which we haven't yet found out why it's in males more than in females. And really, what is it that's transmitted from, you know, the parents? What genes really predispose you to that?
Here the point is to try to understand if this means that these modern humans, our ancestors, were cognitively different from Neanderthals or not. I mean, to be able to paint this thing, it maybe means that you have a different brain or different structure in your brain, but maybe also means that you have, say, something like a spare time. I mean, for being painting here, you must say, you know, someone must go hunting, you know, for you. I'll say, yeah, just go, I need to finish my lion. So maybe what we are seeing here is just simply a different, say, social structure among Neanderthals and modern humans. Maybe the modern humans were larger groups.
Social brain. Tell me what we mean by that.
As you pointed out very nicely in your introduction, we are immensely social beings. We use our social behavior to find a partner, to build a family, to build a community, to build our culture.
Those creatures on Earth who reached a very high level of social organization.
That'd be us.
That's us.
Right.
That's among the big animals.
Right.
And the social insects,
right,
and a lot of other small creatures that reach very high levels of social organization.
That'd be ants.
Bees, wasps, ants, termites, and then there are a few others, aphids, and so on, that became highly social. Have pretty well taken over the Earth.
And by highly social means simply the capacity to live together in groups?
It means living together in groups. We use a special term, Eusocial, right. EU, which means good or solid or secure. And we save that word for the societies in which there's a lot of cooperation and some division of labor so that few individuals live longer and reproduce more than the others as part of the social organization. And that's what the ants and bees, wasps, and humans, and a couple of other mammals do. And that's a very interesting phenomenon because it raises the question, if high social organization, which we manifest more than anything else, is so successful, why isn't it more common? So I've raised the question as in that form, and I've also said, why did it take so long to evolve? It took humans most of the history of life, which is over billions of years before we appeared, and the ants, the bees, and the wasps that dominate. I mean, they make up most of the insect biomass, just sheer weight of insects. They didn't appear until, the early age of dinosaurs. It took forever. So the question is, what took so long? And that has led me then to a whole new layer of investigation in which I set out to find out, how did it happen, this thing that took so long?
And? How did it happen?
Well, it turns out that we just haven't been looking in all of our arguments about what's the main force, what's the main circumstance. It preoccupies anthropologists about humans and the like. We should have been looking more carefully, first of all, at all the social organisms in the world to see if there's a general law, just the way we look at gut bacteria.
Right.
To find out the basic of molecular genetics, just as we look at these little roundworms to figure out how nerve cells put together in a brain.
This week we're happy to have Gregory Cochrane on the show. He's the author with Henry Harpending of The 10,000-Year Explosion, How Civilization Accelerated Human Evolution. The primary proposition that it calls into question is that human evolution itself has stopped. We were taught, I was certainly taught, that once humans developed culture, they, in essence, figured out a way to avoid the pitfalls of natural selection. Cochrane and Harpending say this is not true, that there is good evidence that humans are evolving in the ordinary natural selective way, and that they have been doing so quite rapidly in recent times. And one of its primary theses is that it was agriculture that created the conditions for additional evolutionary pressure. That prior to agriculture, humans had to do one set of things, and then after agriculture, they had to do another set of things. And in between those two moments, it began about 10,000 years ago, a kind of intense period of natural selection took place.
We should be looking at all of the different species of animals that have made this huge leap up in evolution to find out what they held in common. And in a nutshell, it is this, and it explains the rarity and why it took so long. They have to go through a series of steps leading up to the threshold before they go from solitary to advanced social behavior. And these steps include one common feature for all of them, and that is the final step is, and it's a solitary species, a lot of individuals that are solitary, live together, forms or creates an expensive and valuable nest from which it forages out, gets prey and other food, and brings it back, feeds the young until they mature. Everyone, without exception, of these species that made the jump over into eusociality, including human beings, had to go through that process.
Maybe you could describe exactly your argument about how agriculture changed the human genome.
Well, we think that as a consequence of agriculture, population goes up on something on the order of 100 times in the places where it's adopted. People start living in villages. People are sedentary. People start having, more complicated societies where, you have upper classes that live off the surplus or the extracted, not very voluntarily, from the other people. You start having, some cases you have fairly strong governments. You used to be maybe a little hungry as a hunter-gatherer. Now you're a little hungry as a farmer because population has increased to meet the food availability. They're shorter. They're like, in the Middle East, people got shorter by something like 5 inches. I mean, it's only recently that people are finally getting as tall as they were back before agriculture. It wasn't a very balanced diet. But there's some evidence that a lot of genes, new versions of genes that help you deal with this diet spread. The most famous is lactose tolerance, because there was a new food that was available. I mean, it would have been hard to make a living off lactose if there weren't any domesticated animals, unless you had a whole lot of wives or something.
Yeah, I see what you're saying.
But after people domesticated cows and goats, there was this potential source of food that, by the way, kids could use and probably did all along. Like, suppose you had some kid, his mother dies or they're a little short on milk. I'm sure they were giving them cow's milk before people were generally lactose tolerant, because small kids are always lactose tolerant. The difference is when if you keep this ability in adulthood. But that spread, that was very favored when it happened, and spread. There's a version that spread over much of Europe and down into India.
And so how do you determine who does it and who doesn't do it?
That's the accidents of evolutionary history. We should look at evolution sort of like a maze. You start, say, as solitary, and you evolve. Your species, you evolve. And at some point, the environment is changing or opportunities arise, and you take a turn. You move in a certain direction in evolution.
But there's another type of evolution that we need to know about and that's starting to become more important. And that is convergences, where two organisms come together to make one. And of course, in a sense, we're an example of a convergence. I mean, we had the sperm from our father and the egg from our mother, and we're the descendants, so we really have to have genomes coming together to make a new type of individual. But this is done time and time again in evolution, but it's not studied. The reason is that it's very complex. We were sure, absolutely sure, that when we got the first genomes, that these would tell us about, do the tree of life, we'd have every little bit of life understood, there'd be no problem. It didn't happen. We, in fact, began to wonder, and different people proposed, well, we have horizontal gene transfers, and of course that was part of what our lab proposed as well, that individuals, when they live together, they start to exchange genes, so that there's a really complex arrangement of gene changes. That's, in fact, when we looked at the genes from various genomes, we find out there's no single tree, that every gene tells a different story.
If one person carries some things from Neanderthals, those things will show up as cases where a person might be very different from another person, but more similar to a Neanderthal. That process has really just begun. So we can show that when we compare two people from Europe, their whole genomes will have certain elements in them that are quite different from each other, where one person is identical to the Neanderthal genome, and the other person is basically like people who today live in sub-Saharan Africa. So that's the direct sign of this introgression of genes from Neanderthals. But how do we look for it? What do we actually see when we look at the functional parts of genes? My idea from before we had Neanderthal genetics was that it was likely that we would get genes from Neanderthals just as a function of them being relatively closely related to us and the possibility of interbreeding happening. And it turns out that that idea has been carried out. We did get something from Neanderthals. But when we look at the way that this transfer of genes has been studied in the history of biology, it's a process called introgression. You're getting genes from one population and introgressing them into another population. This was studied during the 1940s and 50s in great detail by looking at plant breeding. And the idea was that there were traits in plants that might be adaptive, that were spreading from one population to another. Edgar Anderson, the botanist who studied this in the most detail, said that introgression from one population to another is something of a paradox. It's a paradox because the more important it is, the less likely you are to notice it. And if you think of this, if you were looking at two kinds of plants and you could really differentiate them very clearly, but you noticed that once in a while there was a flower color in this population of plants that was actually from the other population, or that there was a seed shape that had transferred, you would notice it because it sticks out. It doesn't belong there. But when you look at things that are much more widespread across the genome, introgression might be more important, but it wouldn't be nearly as noticeable to you.
When you talk about altruism, what do you mean by it in context of, take the ants?
Well, in biology we use it in a very special way, and that means that you give up some of what we call genetic fitness. That is to say, you give up some of your expected longevity, or the number of children you're likely to produce, or both, in the service of others in the community. And that's what we call altruism of the eusocial type. That's how these advanced societies get bound together by that type of division of labor based on altruism.
Is there a gene for altruism?
We probably is, but we haven't. We know they're there. In human beings, for example, we know from twin studies and other evidence that there is a genetic basis for empathy.
I'm going to ask a sort of almost unthinkable question now, which is whether 0 degrees of empathy is necessarily always bad. There's at least one condition in psychiatry where individuals struggle with empathy, but it doesn't necessarily lead to acts of cruelty, and that is people on the autistic spectrum. So here we can see people with autism and the related condition of Asperger's syndrome. They have difficulties in social relationships and communication, and they too struggle to empathize. Their low empathy tends to lead them to avoid other people because they find people confusing, to withdraw from the social world rather than to commit acts of cruelty. So this is telling us that, that low empathy may be part of what happens when someone commits a cruel act, but it's not a symmetric relationship, that if you have low empathy, it doesn't mean you're necessarily going to commit a cruel act. People with autism process information very differently to other people. In particular, they've got strengths in certain ways of seeing the world. They tend to notice small patterns, small details that other people miss. And the reason that I call them zero positive is that alongside their difficulties with empathy, their unusual attention to detail often leads them to giftedness.
I like this quote. If by some magic autism had been eradicated from the face of the earth, then men would still be socializing in front of a wood fire at the entrance to a cave.
Because who do you think made the first stone spear? It was an Asperger guy, and if you were to get rid of all the autism genetics, there'd be no more Silicon Valley, and the energy crisis would not be solved.
And people vary in the degrees of empathy that they are capable of showing, just like they vary in degrees of just about all their emotional responses and various abilities.
Social behavior is so important that it is conserved in evolution. And you find it not only in people, but you find it in simple animals, some of which, like ants, build complex societies.
A team of scientists led by Ehab Abuhaif of McGill University has unlocked a pattern that explains some anomalies as the reemergence of ancient ancestral traits. Through their work with diverse ant species, Abuhaif and his team reveal that super soldier-like traits can lay dormant and be induced by exposing larvae to an overdose of a naturally occurring hormone. Abuhaif spoke with me about the work and began by explaining just how the ant caste and subcaste system works.
Usually you find a single worker caste, but on several occasions the worker caste has expanded and diversified into multiple subcastes. For example, in the hyperdiverse ant genus Phaidole, all species have a minor worker caste that basically forages and takes care of most tasks within the nest, and then soldiers that actually act in defense. And in other ant species like army ants, you've got as many as 16 subcastes.
And then one of these subcastes is even referred to as super soldiers. What's the story there?
The reason I refer to this ant genus, Phaidole, as a hyperdiverse genus is because it's one of the most speciose genera, period, of all animals. And what's thought to really drive this diversity, of course, is the evolution of these discrete subcastes. So in all Phaidole you find, with one exception, a minor worker caste, which is quite small head, and then a soldier caste, which is larger than the minor workers and has a rather large head and sort of mandibles. But very interestingly, in southeast Arizona and northern Mexico, out of about the 1,100 species known of Phaidole, there are 8 species that evolved exclusively in this region and are known as super soldiers because they essentially look like soldiers, but they're much larger, and they even have larger heads and a larger body, and they have massive mandibles. One of our collaborators in this study, Ming Wang, he described their behavior as such that they use their large heads to block the nest entrance when attacked by army ants.
Okay, so some species here have this subcaste of super soldiers. What about these other species that don't? Do they have any other sort of anomalies within their development?
Well, this was one of our surprises. You know, I've been collecting Phaidole in Long Island, New York, and none of them have a super soldier caste. And one day we realized, oh my God, there's some monstrous-looking soldiers there. And when we picked up this anomaly we were looking at, we realized that it looked surprisingly similar to a super soldier subcaste that evolved in southeast of Arizona and northern Mexico. And the other thing is, too, they had these little wing vestiges on them. I mean, there's not a single species of ant in which the workers have wings. They look so similar to these super soldiers, it got us digging a little deeper, you know, into their origins.
Right, and so this is what you were most interested in, was then, you know, how these super soldier subcastes evolved, and then also how these anomalies that were super soldier-like could have also evolved in different species. So what did you find?
Well, there was a couple of things. We started to try to piece together the puzzle as to what this similarity is telling us and how you could get these anomalies being produced. And so we took a very developmental approach to this and just said, well, if we could figure out how this super soldier anomaly produced, I think we could figure something about the evolutionary developmental origin of both. When a queen lays an egg, if it receives the right sort of photoperiod and temperature cues, it becomes a queen. If not, that egg develops into a worker. And then based on nutrition, during the last larval instar, if it gets the proper nutrition, it develops into a soldier. If not, it develops into a minor worker. And these switches during development are governed by a hormone called juvenile hormone.
So this is where we get to the research on fetal testosterone. Although people think of it as the male hormone, both sexes produce it, but males produce a lot more than females, and this is true even at the fetal stage. Testosterone seems to have organizational effects on brain development. In humans, as well as other animals, the hormone does its work by binding to androgen receptors, which are all over the body, but are also found in the brain. And it's a good candidate for being a biological mechanism for influencing brain development because it affects neurotransmitters like serotonin and GABA. And you can see again, girls have a much larger vocabulary than boys at the age of 2. And this has been known about for a long time, that girls talk earlier than boys. Boys are overrepresented in clinics for language disorders like stuttering. The higher your testosterone level prenatally, the smaller your vocabulary at 2 years old. And I should say here that these results hold up when you combine the sexes, but also when you just look within one sex, let's say just within the boys, suggesting it's not just redescribing the sex difference, but it's actually a hormone effect. Jumping ahead now, these children are 8 years old. We can give them that eyes test of empathy. And once again, this line slopes downwards. So the higher your fetal testosterone, the more difficulty you're having in reading emotions from the face.
Being social beings, we need to recognize each other. And we do so in all sorts of ways, by the way people move, the way they dress, their voices, where they are. But in particular, we are very good at recognizing each other's faces. Well, when I say we, I mean the majority of people. There are some people who are not so good at recognizing faces, and they are called face blind. They may see a face perfectly clearly, but it doesn't convey identity to them. And the technical term prosopagnosia, prosopon is the Greek for a face, and an agnosia is not recognizing. So the term prosopagnosia was introduced in 1944.
We may take facial recognition of our fellow humans for granted, but not all species have evolved such a specialized trait. Indeed, a duo of scientists at University of Michigan, Ann Arbor has zeroed in on 2 closely related species of paper wasp, one that possesses specialized face learning and another that doesn't. A series of experiments involving photos of wasp faces as well as general shapes and patterns indicates that while one species does recognize faces much more readily than the other, recognition of faces develops separately from other visual and cognitive abilities. The researchers also argue that the capability evolves independently across varying animal lineages. Lead author Michael Sheehan spoke with me about the work.
So the research shows that one particular species of paper wasp, Polistes fiscatus, which we have previously shown recognizes individuals, does so through a process which is relatively similar to how people recognize individuals using their faces.
Although one talks about face blind as if the face blind are a species apart from the face normal people, in fact there's a whole population, really a continuous population, with the 80% of the people in the middle, and the poor recognizers at one end, and the super recognizers at the other. The super recognizers are people who have indelible memories of faces, who will say, yeah, I remember you, you served us in that cafe in Naples 27 years ago. I think my mother was probably bad at recognizing faces. She was very shy, she was ill at ease with large numbers of people. My father was the opposite. My father was a sort of super recognizer. I think he recognized thousands of people, and he was highly sociable.
So this one species of paper wasp has specialized face processing. They learn faces differently than they learn other types of images. But closely related species of paper wasp, for example, Polistes metricus, is unable to do that, and they don't recognize faces at all. In fact, have a very difficult time telling faces apart from each other. So what we show here is that specialized learning in wasps and people have evolved in parallel in these really different taxa, but in closely related wasp species, we find really different abilities for learning.
You find that there are a number of areas in the brain that respond much more strongly to faces than they do to objects. This is one example here. This is the most well-known of these areas. It's called the FFA, the fusiform face area. So it's an area in the fusiform gyrus. This is actually in the right hemisphere, and it's usually much stronger in the right hemisphere than in the left. These are areas that are often compromised in prosopagnosics.
Now, Nancy Minshew did another brain scan, and she found I was more interested in things than I was in looking at pictures of people. But the thing is, we need to have people in this world that are interested in things.
If you look at the birth of Apple, the Apple computer, we tend to associate Apple with Steve Jobs, who was this dazzling showman. But really, the person who invented the Apple computer was a different Steve. It was Steve Wozniak, who was a self-identified introvert. And he created this amazing thing by sitting by himself in a cubicle at Hewlett-Packard, where he was working at the time, late at night and early in the morning before anybody was at work. He would work by himself for months, and then he produced this amazing thing that he then shared with his friend Steve Jobs.
Dr. P, whom I described, had overwhelming problems with agnosia of many sorts. He didn't recognize people, although interestingly he recognized cartoons, because in a cartoon there is an exaggerated nose or an umbrella. There are particular marks by which one can recognize a cartoon.
You know, the way I work is to make this kind of Brobdingnailian world in which I make the face into a landscape. And I journey across that landscape like Gulliver's Lilliputians, crawling over the face of a giant, not knowing that they were on the face of a giant, but knowing everything about that face. And then what I do is I put all that information together, or the kind of nose, the nostril, the corners of the mouth, whatever, and I can commit it to memory. So I know that it's no accident that I was driven to make portraits of people who matter to me.
I think I also have a vivid love of humanity, as Chuck has, but for me the portraits take the form of narratives, of stories.
Yeah.
Although I'm not very good often on details of what people look like. The first time I took a piece into the New Yorker, the then editor, Robert Gottlieb, this was about an Italian painter, he said, well, what does Franco look like? And I said, you know, the piece is about his work.
Well, your words paint the most specific portraits of people's lives, and I always have identified your characters as vivid a portrait as anything anybody could do in any other art form. And I mean, you know, you celebrate your connectedness with humanity in a really important way. I mean, it's a way that we can identify with, even if it's a problem we don't have. Empathy is the basis of, I think, the mortar that holds society together.
What about systemizing? You know, does your child like to collect the whole set of playing cards or toy vehicles? Or how interested is the child in different types of toy vehicles, the make and model of different cars, and so forth? The higher the child's fetal testosterone, the stronger their interest in systems at aged 8. So we can look at autistic traits, and this it addresses this relatively new notion, namely that autistic traits may be distributed right through the population. We all have some autistic traits, is the idea, and that people with a diagnosis are shifted to the right, so they have more autistic traits, but this is the general population, and so we've given that to these children, and again you can see a positive correlation that the higher the child's fetal testosterone, the more autistic traits the child has. Now that these children are old enough, we're also getting them to climb into the MRI scanner, because the hormone testosterone affects brain development. So we picked out the corpus callosum as our first region of interest because of the sexual dimorphism in the general population, to say, is volume in any of these subsections of the corpus callosum related to hormone levels? And you can see the way that you section the corpus callosum, there's a positive correlation with asymmetry of just this fourth region within the corpus callosum. But this is just the first of a series of studies looking at the correlations between hormone levels prenatally and neuroanatomy.
So how does all of this tell you about how these super soldier ants evolved within this genus?
Well, two things. So then we look for two species, Pheidole obtusospinosa and Pheidole rhea. These are two species which have naturally evolved super soldiers. And they make up about 4% of the colony, and they have a function in defense. We figured if we look at their development, we should find that relative to the soldiers, they should also have an elaborated size during development and also an elaborated wing disc pattern, if our predictions are correct in terms of development. And that's exactly what we found. And so that was the first part of our prediction. And then we went on to try and figure out, well, okay, it's really odd out of this 1,000 species that all of these super soldiers would just occur in one region. So it was likely that they just sort of diverged from a common ancestor. But then we were surprised again because when we reconstructed their phylogeny, these two species that we had sampled are actually quite distant on the tree, because using the same developmental programs to evolve things in very different parts of the tree on separate occasions.
And you, in your paper, you bring in the term genetic accommodation here, or genetic assimilation. How did that play a role?
What tipped us off is some of these pioneering studies when they were first figuring out, how these switches are governed. And they discovered that it was juvenile hormone. When they were applying the hormone, they always could switch between minor worker and soldier. And there was something interesting sort of in the background of those studies that whenever you used slightly higher dose of hormone, you would produce slightly larger individuals. And so we went to the same species where we found the anomaly, and we applied hormone during a critical period in development, slightly higher doses. And then, amazingly, we were able to induce super soldier development. And these are species, of course, that lack a super soldier cast. They don't have them. And, of course, interestingly, when you induce them, you also induce the development of really large larvae, like you do in the natural case, and you also get these massive vestigial wing discs. You are also turning on the same kinds of genes. And so really we were convinced that you're really inducing sort of a dormant program, that there's a potential there, and then when the need arises, and somehow these super soldier anomalies are beneficial to the colony, selection can actually fix the constitutive expression of these anomalies and make them real. And that is a process of genetic accommodation.
So what is the significance of this finding in the bigger picture of evolution?
What we found, essentially, is something very general. Going back to Darwin's time, and even before and after Darwin, phenotypes that get induced in populations that reflect these kind of ancestral traits, they're usually called atavisms, but we see them being induced in populations all over the place. And previously, evolutionary biologists had treated them as sort of the Barnum and Bailey of evolution, or some kind of slips of a developmental system. And what we're showing here is that no, if you were to look at the tree and see that super soldiers are popping up all over the place independently, we would have thought, okay, they evolved de novo, they evolved basically the super soldier caste independently several times. But the fact that we can induce it in species that don't have it all over the tree reflects some deep underlying potential that goes back to the ancestor. So the ancestor must have had this trait. It lost the expression of the trait, but it retained this genetic potential. And this genetic potential lied dormant, at least in Pheidole, for 35 to 60 million years, showing that potentials can be locked in place for a very long time. And so we think that this is not specific to Pheidole. All over the five kingdoms, you see these kinds of potentials just kick back in. We've really not given them attention, but I think they hold enormous power for evolution. And this is something I think once our study gets published, I hope that people will be looking for these hidden potentials and their power for evolution in other species.
How would you hypothesize that this this difference would play out in the phenotypes of boys and girls with autism, both in their characteristics, their autism-related characteristics, and also in their developmental patterns?
What we're expecting is that girls may be masculinized by elevated testosterone levels. And well, you're asking about the phenotype, so we're expecting that wherever you find sex differences in the general population, like in language or patterns of interest, like play, looking at faces, these kinds of things, where you do find sex differences, we should expect an absence of those sex differences in autism, that the girls may become much closer to the boys, if you like. It's not necessarily going to be the case that there'll be no sex differences even within the autism group, but I guess the prediction is a reduced pattern of sexual dimorphism. And we're also wanting to test that at the level of the brain. We were discussing this before the lecture, that we're starting a study of structural MRI study and DTI study of adult women with Asperger's, to look at areas of the brain which are normally sexually dimorphic, to see whether those are absent when it comes to comparing males and females on the autistic spectrum.
And this is what we do have right now. We have the structure of age and sex of 12 Neanderthals individuals that, remember, apparently were a family group. We have 6 adults, 3 males and 3 females. We have 3 adolescents, 2 juveniles, and 1 infantile. And I got now mitochondrial DNA from all these individuals. So therefore, we can have information on the diversity within Neanderthal groups. We can have information on kinship structure and also on mating behavior, as comparing, for instance, mitochondrial lineages in males as compared to females.
We know that we have common ancestors with Neanderthals, but in addition to that, we know that we have Neanderthal ancestors, because our genetic makeup is in part their genes, with about 3% contribution to everybody living today outside of Africa, and possibly a smaller contribution inside of Africa. So we know that we've got their genes. What we don't know is whether those genes do anything interesting. Is there something that we got from them that we wouldn't have gotten if they weren't our ancestors? The first way that we might think about this question is by thinking about the one gene that we know the most about, and that's the mitochondrial DNA.
And now for what is, totally objectively speaking, of course, the coolest part of the animal cell, its power plants, the mitochondria, the smooth, oblong organelles where the amazing and super important process of respiration takes place. Mitochondria are in the egg cell when an egg gets fertilized. And those mitochondria have DNA. But because mitochondria replicate themselves in a separate fashion, it doesn't get mixed with the DNA of the father. It's just the mother's mitochondrial DNA. That means that your and my mitochondrial DNA is exactly the same as the mitochondrial DNA of our mothers. And because this special DNA is isolated in this way, scientists can actually track back and back and back and back to a single mitochondrial Eve who lived about 200,000 years ago in Africa.
Well, as I mentioned, we know more about the mitochondrial DNA in Neanderthals than we do about the rest of the genome because we have a larger sample of individuals. We have more than a dozen Neanderthals from whom we've got complete mitochondrial DNA sequences. Their mitochondrial sequences are all distinct from ours. No living person carries, as far as we know, a mitochondrial sequence that looks like it came from a Neanderthal. So when we talk about the Neanderthal contribution to our DNA, this is a contribution that we've evaluated by looking at other parts of the genome, the nuclear genome.
But of course, for understanding what happened when modern humans met Neanderthals, this question is how did we interact with each other is rather important. Because after all, when two groups meet each other and there's some social inequality, one almost always mixes, but it's generally rather directional. The offspring of such unions will generally stay with one or the other group.
We don't see the signal of our inheritance from them in the mitochondrial genome. The mitochondrial DNA is in some sense following a different kind of history than most of the genome. And if we can show that it follows a different kind of history between humans and Neanderthals, well, that's telling us that the mitochondrial DNA must itself have been affected in its evolution by its function.
This is a rapidly evolving field, but I'll say in the outset that really over the last probably 6 months, it's been clear that we can distinguish primary mitochondrial disease and secondary mitochondrial dysfunction. And it really, the vast majority of children with autism spectrum disorders will not have a primary mitochondrial disease defect. And the remaining may have epigenetic forms of mitochondrial dysfunction. I'll say that word again, epigenetic, because I'm going to finish my talk with that word, and how what the implications for the interaction between our ancestrally transmitted gene set, that we've received from our ancestors who had to interact with their environment, the foods in their environment, and the microbes in their environment, the viruses and bacteria and fungi in their environment. So mitochondrial disease and autism are not the same. Autism is really a triad of disorders involving neurodevelopment, brain, GI tract. Oh yeah, so I should make that point right away. So from an evolutionary point of view, there's an argument to be made that our brain is actually our second brain.
So my story is a tale of two brains, because it might surprise you, our gut has a full-fledged brain. All the managers in the room say, you don't tell me something new, because we know, gut feeling, this is what we are using. And actually you use it, and it's actually useful, because the gut is connected to our emotional limbic system. So they do speak with each other and make decisions. But what it means to have a brain there is...
during the Cambrian and the development of the first multicellular organisms, imagine the sea anemone, where the coordination of its GI tract was regulated by neurons and by actual neurotransmitters and peptides that eventually became co-opted with evolution to a site of greater neuronal development in what we call today our brain. Okay, but the gut, from an evolutionary point of view, was our first brain. And our brain is really our second brain, and that's why there's so many connections between the two, is because they're father and son.
Scientists have recently shown that bacteria in our gut, or in rats' guts, can influence not only brain chemistry but behavior itself. There's certain gastrointestinal diseases, which include irritable bowel syndrome, which is fairly common, that are often associated with depression or anxiety. And there's also been some hypotheses put forward that some psychiatric disorders, Steve, have you heard of this, like late onset autism, may be associated with abnormal bacterial content in the gut.
Yeah, I think that's speculative.
That's why I said it was a hypothesis.
Here is a model of a wall of the gut. I took the three elements, stomach, small intestine, colon. And within this structure, you see these two pinkish layers, which are actually the muscle. And between this muscle, they found nervous tissues, a lot of nervous tissues, which penetrate actually the muscle, penetrate the submucosa, where you have all the elements for the immune system. The gut is actually the largest immune system, defending your body. And how this brain takes care for this, to moving it with the muscles and to defend the surface. This brain, which is autonomous, has 500 million nerve cells, 100 million neurons, so around the size of a cat brain. So there sleeps a little cat. Okay. Thinks for itself, optimizes what Heribert digests. It has 20 different neurons types. So the same diversity you find actually in our big brain, where you have 100 billion neurons. It is autonomous, organized in microcircuits. It has these programs which run. It senses the food. It knows exactly what to do. It senses it by chemical means, very importantly by mechanical means, because it has to move the food. It has to mix all the various elements which we need for digestion. This control of muscle is very, very important, because, there can be reflexes. If you don't like a food, especially if you're a child, you gag. It's this brain which makes this reflex. And then finally, it controls also the secretion of this molecular machinery which actually digests the food we cook.
Animal mitochondrial biology is is what I call the secret life of mitochondria. In my view, the cellular defense functions of mitochondria were actually more ancient. With the first eukaryogenic event that occurred
long ago, they existed as their own sort of bacterial cell. And one day, one of these things ended up inside of an animal cell, probably because the animal cell was trying to eat it. But instead of eating it, it realized that this thing was really super smart and good at turning food into energy, and it just kept it. It stayed around. And to this day, they sort of act like their own separate organisms, like they do their own thing within the cell, they replicate themselves, they even contain a small amount of DNA.
And for single cells can do just fine on glycolysis. They don't need mitochondria for energy production. Okay? You just don't need it. It's, really you need mitochondria for energy production when you start putting multiple cells together to make, animals and plants. There is a different mitochondrion specialized for every one of those cells. Okay? So they are adapted to meet the metabolic demands of each individual cell type. Cholesterol, cortisol, steroid hormone synthesis, vitamin D activation, inactivation, and the first committed steps of innate immunity.
Professor Parham's team has been comparing the genomes of those prehistoric people with the DNA of modern humans. They found that the Neanderthals and Denisovans had particular gene variants, or alleles, that play a big role in immune defence.
So from both of these archaic samples, it looked as though there were alleles that came into the modern human population in Asia and in the Middle East, and then spread out as human populations migrated and increased in size.
He says it was gaining those alleles through mating that would have helped our ancestors thrive. But Professor Parham warns there could also be a downside.
The downside of having this new haplotype that came from somewhere else was that it gave a greater risk of not being able to control the end of the response to infection and leading to autoimmunity.
He says that could explain the increasing prevalence of diseases like lupus, celiac disease and autoimmune hepatitis, where the immune system actually starts to attack the body.
Researchers studied the medical histories of children born in Denmark over a 10-year period. Children with mothers who had celiac disease, a digestive disease where people cannot tolerate gluten, were nearly 3 times as likely to be diagnosed with autism. A family history of juvenile diabetes was also associated with a higher risk of autism. The risk doubled for children whose mothers had this form of diabetes. And children with mothers who had rheumatoid arthritis were also at a 70% increased risk for autism. This suggests that prenatal environment may be affected by certain maternal autoimmune diseases.
When a cell is in an environment that meets all of its nutritional and physical needs, mitochondria will produce a set of small molecules that get sent to the nucleus and will get sent outside the cell that participate in, that actually create a little halo around the cell of high concentration, and that halo glows with a color that says, "I'm well." Okay. And when the cell finds itself in a hostile environment, when it's exposed to any environmental chemical, whether it's mercury, cadmium, or this is polybrominated diphenyl ethers as flame retardants, you will produce a change in the metabolic function of the mitochondria and will change the small molecule words that they use to communicate with the nucleus and neighboring cells. Oh, and the one that frequently gets omitted is meiotic recombination. So if you pull out the mitochondrial DNA, one of the most profound defects that you observe is that now those cells cannot sporulate. They cannot produce gametes. Okay. It also seems to be that, at the heart of why a number of hybrid crosses, for example, between a horse and a donkey, give rise to a mule that is sterile because of the incompatibility of the mitochondria carried from a horse, with the nuclear DNA, carried from the donkey. Okay, there are lots of examples of these things that are kind of arcane.
And when we compare the actual gene sequence of the mitochondrial DNA one of the coding loci, one of the genes that has a function which lies on this DNA, is COX2. COX2 is essential for oxidative metabolism in your cells. It's one of the things that makes energy for your body. And that gene, with mutations between the human and Neanderthal version, apparently had a somewhat different function in Neanderthals. It looks like the mitochondria of Neanderthals may have been adapted to a different energy environment than the mitochondria of living people. That's probably not surprising, because if we think about Neanderthals living in a Pleistocene environment in the cold, temperate, and subpolar parts of human habitation at that time, with a very limited technology in terms of clothing, and tight clothing in particular. They didn't have that stuff, and they did eat a somewhat different diet from recent Europeans. Put all those things together and you can imagine that their mitochondria might have been adapted to a very different energy scenario than the mitochondria of more recent people.
Okay, so the evolutionary origins of this, I'll just briefly go over it. So our ancestors had to deal with seasonal differences in availability of calories. In the winter, calories were limiting. That's on the right-hand column here. So calories were limiting. You had to work hard in order to get adequate calories. Okay, and there are certain hormones that were associated with low-calorie existence. This is a time when the tortoise wins out. Okay, one of the problems of human disease now is we're in an eternal summer, an endless summer. Okay, we don't have cycles of nutrients anymore. Okay, through winter and summer. So anyway, so that's kind of a little brief overview about the summer and winter functions of mitochondria that are played out in another scale during the day when we eat and the night when we don't. There are smaller variations in that. And that's also associated with small variations, circadian variations in NAD and NADH, and as well as circadian variations in cholesterol biosynthesis, etc. So I'll leave it at that.
If you today have your genes tested so that you've sent your DNA away to a company and they've given you results about this, one of the primary things that they're likely to tell you about is where in the world people who have similar mitochondria to you live today. Where did your mitochondria come from is sort of the question that they're likely to be able to address. And so you may be looking at a map, and that map will show you that your mitochondrial haplotype is found in a number of different areas of the world. Some of them, it's really common. Other areas, it may be more rare. But there will be a distribution where you've got your mitochondrial type shared in people that are living in the world now. That can be exciting to look at. If we did this exercise for Neanderthals, the map would be blank. There's nobody living in the world today who has their mitochondrial type. So that avenue is a dead end.
So autism is kind of interesting. It's a moving target. If you go back and you plot the prevalence of autism dating back to 1990, and you can actually create a very nice exponential curve that goes through about 2005, with 18% growth per year. 80% of the patients in the U.S. with autism spectrum disorders are under the age of 22. So what about the epigenetics of mitochondrial DNA? So we evolved in Africa, and then our ancestors moved to the Middle East and Northern Europe and Asia and crossed across the Bering Sea into the Americas. That all occurred tens of thousands of years ago. Okay, but our ancestors dwelled and became part of that land, and we learned to live with the nutrients in that environment. We learned to survive in the presence of the different microbial environment. But what happened recently in the last 300 years is that we had this massive exodus from Northern Europe and from Africa to North America, and that brought in genes that had all co-evolved with their environments, for thousands of years earlier, into a new environment with new food sources and new viruses and bacteria and fungi to evolve with. And then, just as we get good at that, then we change the game again and start, with dramatic changes in the way that we do farming, in the way that we do our fishing, where concentrated animal farming operations, really are producing more and more of our food supply, and we're using the powers of our brain to generate chemicals that our environmental history, our evolutionary history has never encountered before. And so we're having to readapt, okay, to this changing environment.
Three great unknowns in biology and human social biology. The genome, we have lots of sequence, but we really don't have any idea how it works. The human brain, probably the most complicated structure in the universe. We certainly have no good idea how it works. And the evolution of modern humans. What are the selective pressures that have been operative in producing us after our divergence with chimpanzees about 6 million years ago? How can we study these great unknowns, preferably together? I've chosen to do so by focusing on what I call cognitive and psychiatric conditions. And the basic logic here is that in the same way that one studies the normal adaptive function of a protein or a gene by perturbing it or knocking it out, one can study the normal functions of the human brain by looking at various forms of neurocognitive variation that are commonly referred to as disorders. The causes of this neurocognitive variation can and certainly have been traced to genetic and epigenetic variation in neurological function, so we can connect the conditions to their genetic and epigenetic underpinnings and their neurological correlates. And this genetic and epigenetic variation can be traced in human ancestry via molecular genetic studies. So this work involves integrating evolutionary genetics with molecular psychiatry, the neurosciences, and psychology behind enemy lines, as it were. And to do so, we need to connect evolutionary genomic variation with variation in neurodevelopment, cognition, and behavior. We need to connect across diverse, formerly highly diverse disciplines.
Is evolution slowing down in humans? Or is evolution continuing or even speeding up in our species? This is a part of our prehistory that I have spent a lot of time researching. My early genetic researches looking into the patterns of recent natural selection in humans has really led to some insights about the pace of recent evolution. And when I say recent, I really mean sometime within the past 10 to 15 to 20,000 years. In other words, the final sort of chapter in our evolution associated with the development of agriculture and the spread of humans throughout the world. So we're looking at that very thin part of our evolution at the end of this long process that we've come through to this point in the course. We've known for a long time that during the course of the last few thousand years, there have been biological changes in humans. We see those changes in the skeleton, and they were described in the last lecture. The kinds of changes in the shape and size of the skull, those are really evident when we look at skeletal samples of people from before the development of agriculture, from around the time of agriculture, and in more recent samples of people of agriculturalists. But what has become clear in the last few years is that these changes in skeletal form are accompanied by other massive changes in genetics. A very good example of this is a gene called lactase. Lactase is an enzyme that allows you to digest a sugar called lactose. And that sugar in nature really naturally occurs only in milk. So it's a sugar that if you're not a milk drinker, you don't have access to. Well, of course, every human is a milk drinker sometime in his or her life, just as all mammals are, because milk is the natural nutrition that babies get and that infant mammals get when they're born. But most people, and essentially all other kinds of mammals, lose the activity of this gene after they develop past weaning, after they stop drinking their mother's milk. And the reason is quite natural. If you're a bull, a full-grown male bull, you don't nose up under the cow for milk. It's not a food resource that you're going to have available to you. And so why make the enzyme that's only necessary to digest this particular kind of sugar? Most mammals don't do it. They turn it off. But some humans leave it on. They continue to make that enzyme as they're adults. And the humans that leave it on, we could call them mutants because they actually have new mutations that keep this enzyme active. That mutation, which has occurred 5 different times in 5 different human populations and has become common today, that mutation, it's entirely new, and it occurs only in human populations that have a long history of keeping and domesticating dairy animals. It's the environment that they've created, the environment where milk is available to them as adults, that has selected for their ability to continue to digest the milk as adults. This genetic change, it's one of the most rapid genetic changes that's happened in the human population, and it's common today in parts of the world with a history of dairying. Well, that's really interesting, but how do we learn about this kind of genetic change? When a new mutation happens, it happens on a copy of a chromosome. For lactase, that's chromosome 2, and we know the particular points on chromosome 2 where a genetic change will cause lactase to continue to be produced. When a mutation happens on a chromosome, well, there are other genetic variants that occur on the same chromosome as that new mutation. And if that mutation starts to become more common, well, it will carry with it the genetic variants that it's linked to on the same chromosome. Those things come along for the ride. They're what we call hitchhiking. And those hitchhiking variants will be relatively far away from this new mutation the younger it is. And as it persists in the population for longer, the process of recombination, when chromosomes cross over with each other, the mother and father chromosome cross over to create a new combined chromosome once in a while in reproduction. As that process goes on more and more, chromosomes are broken into smaller and smaller parts that are identical from one ancestor. So we can tell when we find a new variant that's linked to a bunch of variants along a long stretch of chromosome that are hitchhiking together, we can tell that that's new and that it has become common recently. And when we find evidence for something that's new and common, that's unusual. That means that that new common thing had some sort of advantage in the ancestral population to the people we're looking at today. That's a pattern of selection that I went studying in the genome for. As soon as we had genetic samples from many, many individuals, we could look through genomes to try to identify cases like this. And what we found was that there were many hundreds of cases where there was a part of the genome that had a new mutation that had become common within the past 10 to 20,000 years. The scope of this effect throughout the human genome is so large that when we think about hundreds of things changing in a very short period of time, that's telling us that every one of those things is something that has come from a new copy and that has involved more people reproducing who carry that copy, or more people dying who don't have that copy. And those kinds of things are found in many aspects of our biology. For example, in Africa today, there's a variant of a gene called G6PD. G6PD is a glucose phosphorus dehydrogenase gene. And that gene, if it's deactivated, gives partial resistance to falciparum malaria. The deactivated version of that gene is something that a lot of people have heard of because it gives rise to a condition called favism, where the person is allergic to fava beans. Well, favism is rare. It's most common in the Mediterranean, where fava beans are naturally grown and where some people carry this gene variant because malaria was common there in the past. But it's much more common in North Africa. And that population, which has a G6PD high-frequency deactivation, that population is one that over the past several thousand years has been subject to malaria. G6PD is one of the fastest growing of the new genetic mutations that's been subject to selection. It's grown to its present frequency in probably less than 3,000 or 4,000 years. Some other genes have been changing for longer periods of time. For example, a gene called DRD4, which is the D4 dopamine receptor, has a version that's longer, that has 7 repeats in its DNA, as opposed to 4 repeats or 2 repeats, which are the more common versions. The 7 repeat version in living people who carry it is a little bit associated with attention deficit hyperactive disorder, ADHD. That association doesn't explain all of ADHD. It's not a Mendelian genetic disorder where you inherit it when you inherit this gene, but it is a disorder that's affected by this gene.
Yeah, we've been looking at one of the genetic variants in ADHD. It's one of the dopamine receptors. It's the D4 receptors. There's probably 5 types of receptors, and this is the 4th one. And there's a genetic variation in it, and we're particularly interested in the variant that's called the 7 repeat. Now, people know that if you have this 7 repeat variation of the gene, your risk of having ADHD is increased. And this has been found in many studies. It's been replicated in many different countries. Not always, but most of the time. What we were looking at is, can we go a bit further and look at how this gene variation relates to clinical outcome and to brain development? So what we find in terms of clinical outcome, I think surprised us a little bit, is that we found that if you have this risk variation of the gene, then you actually had a slightly better clinical outcome. And some other groups have found this and some haven't. So it's certainly not a settled finding. But interestingly, we also find that if you have this variation of the gene, that you have quite a distinct pattern of cortical or brain development, the cortex, the grey outer mantle of the brain. And what you find is that in people who have this risk gene, they show with time a sort of normalization of the early problems they have with their cortex. And this is particularly prominent in a part of the brain called the parietal cortex, which is kind of here on the right side. So that was interesting because in a previous study we'd found that normalization of this part of the brain in general is associated with having a good outcome of ADHD. So it's a sort of good example of how you can link a gene variation with clinical outcome and then in turn link that with brain development.
Now, we like to think that the human species is the most advanced species on the planet, and this suggests that genomic complexity has been critical to our success. Yet the origin of that complexity is not obvious from studying the human genome itself. Genomically speaking, we fall somewhere between a chicken and a grape. And if you thought that the number of bases in the genome was significant, well, I'm sorry to tell you that there's not much difference between the person sitting next to you and a turkey or a lungfish. Underlying the genome's complexity is its ability to create diversity. And this diversity comes in a number of forms. For example, multiple non-coding elements are used in combination to define precise yet adaptable patterns for gene expression. The alternative processing of genes and the use of alternative promoters also creates extensive diversity in the genome. Furthermore, non-coding RNA, which was largely undetected until a few years ago, now plays a central role in the theory of multicellular complexity, carrying out a massive amount of regulation within the cell. So with this complexity in mind, maybe it's no surprise that the coding regions cover less than 2% of the genome. And the hunt is on to find altered sequences that impact normal regulatory functions, to understand how they affect regulation, function, and speciation. And regardless of whether these mutations cause single gene diseases or are involved in complex multigenic diseases, they have subtle and far-reaching impacts that create diverse and unpredictable outcomes. So what do we know about the human genome so far? There are 1,400 elusive disease-associated variants in the genome that have been identified through genetic epidemiological studies known as genome-wide association studies, or GWAS. This is a statistical technique
that compares thousands of patient samples to thousands of control samples and has identified sequence variants associated with the risk of Crohn's disease, heart disease, diabetes, and dozens of other conditions. The vast majority of these 1,400 GWAS variants lie in the non-coding regions of the genome, and they're providing us with enormous opportunities to discover what they do. So throughout this talk, we'll take a look at functional categories of the genome to further explain the steps you might consider to ascertain function at these GWAS sites. Now, probably the biggest impact of the Human Genome Project to date is the ability to compare genomes at the nucleotide level. And that's because mutations in functional DNA are less likely to be tolerated and leave a signature of similarity across genomes. Early on, with only human and mouse genomes available, some of the most important insights were gained. For example, while some genes change rapidly, such as immune, reproduction, and olfaction, other loci containing developmental genes are so highly constrained that no base pair has changed in 75 million years of evolutionary time since the last common ancestor. It's pretty amazing. So how much overlap is there between the human and mouse genomes? At the nucleotide level, approximately 40% of the human genome can be aligned to the mouse genome. These sequences seem to represent most of the orthologous sequences that remain in both lineages from the last common ancestor, with the rest likely to have been deleted in one or both genomes. And today you'll find the genomes of 36 mammals and 10 non-mammalian vertebrates aligned in the genome browsers. You'll recognize that we use genomic diversity to uncover the story of human evolution because it refines the historical account of the early migration of humans out of Africa into the Middle East and beyond. And you've probably read that having the sequence of the human genome available was crucial to the discovery of unmistakable traces of Neanderthal DNA mixed into some modern human populations. So what percent of genomic DNA in people from Eurasia and the southwestern Pacific was inherited from Neanderthals? Between 1% and 4%. But did you actually know that this inherited material is not concentrated in genes? Instead, it's spread somewhat randomly in the non-coding portions of the modern human genomes. And doesn't it make you wonder what it contributes?
Specifically, there are regions of sustained increased density that suggests there may have been a copy number difference between the Neanderthal genome that we have sampled and the reference human genome sequence.
Chromosomes can vary in a lot of different ways across people and within people, and the chromosome is made up of repeats of different structures within the chromosome and even deletions of parts of the chromosome. And there are all sorts of diseases in pediatrics typically where you could be missing a little piece of the chromosome. And there's also very small copy number variants, and then even smaller than that, what's called sequencing of genes, where you just find one base pair. This may be far more in depth than I think you want to go, but the kind of copy number variants that we've been studying, which are just changes in material in the chromosome that are too small to see with a microscope, but can be seen in new ways that have been developed to scan all of your genes, the entire chromosome. And we're not just looking for copy number changes in parts of the chromosome, but we're looking for parts that are very rare, because there's a lot of normal variation, just like people are varied in terms of how tall they are and what color their eyes are. There's a lot of healthy variation between people in terms of these very same variants. So when we find a copy number variant in, say, a group of patients, as we have and are still doing, we first have to look it up on tables to make sure it hasn't been reported before in normal subjects. And this is such a new field that the database for the normal variation is growing all the time. So this is a work in progress.
We actually found several dozen genes that are likely to be involved, perhaps up to 100 different genes, which was quite surprising actually. It had been speculated for many years that autism may be arising due to common genetic variations in the population. But in fact, what we found are several rare genetic variations, what we call copy number variations of DNA found in autistic individuals. The genes that we're finding to be involved, they all seem to work together in a connected pathway. So they encode proteins that are communicating and working together. We've identified about a half dozen genes, perhaps 100 more that may be involved.
Right around the time of the separation, or just before the timing of separation of human, chimp, and gorilla, and we think shortly thereafter, there was a burst of duplication activity. So we have on the order of 3 times the number of megabases that were duplicated in this ancestral branch before the separation of chimp, gorilla, and human. This is interesting because if you look at texts that have been written for the last 20 years on this subject, most people have had this paradox: is why is there so few genetic changes over the millions of years that exist between humans and chimps and gorillas? And that is because most of the studies have been done really on single base pair changes and small insertion deletions. In terms of duplication, it appears we have an episodic burst of activity at a critical time, I would argue, during evolution. All right, so why do I show you all this? Well, this architecture, which is having all these duplications that are spread out, that are large and highly identical, creates problems for our genome. Because they're interspersed, everything that's bracketed by these duplications gets taken along for the ride. And now you have gametes that now have additional copies of genes A, B, and C, in addition to duplication, or have lost. So this creates a huge amount of genetic diversity in our population. And as a result, it also creates disease. And so years ago, we identified these 130 regions, and we systematically have shown by studies of human disease that about 45 of them are important in terms of causing sporadic mutations as well as inherited mutations as a result of this process. And these affect diseases such as autism, intellectual disability, developmental delay, epilepsy, and schizophrenia. So our genome is predisposed to these diseases because of our architecture, essentially, that has evolved.
So we feel that in the last 3 years, we and many other people around the world are onto something by looking at these copy number variants.
These copy number polymorphisms, all of us carry about 10 of them. You can look at families where you have more than one autistic child, and where you think that the parents probably are carrying some genes which predispose the children to autism. You can also do it when 2 seemingly absolutely normal people have an autistic child.
So both of types of differences between humans and chimps, or humans and other non-human primates, and in fact most of the chimp-human differences aren't in proteins. They're going to be in these parts of the genome that used to be called junk DNA. Much of it is doing important things, and so slowly science is starting to understand this non-coding or dark matter part of our genome that used to be called junk. And one of the important things that the non-coding genome does is to control expression of nearby genes. So they're things called regulatory elements, and they can turn nearby genes on and off.
I just want to make a remark. You drew the analogy of the sense of smell, and what a poor sense of smell we have. It's a fascinating fact that although dogs, for example, have a much better sense of smell than we have, as you mentioned, we have the genes that would once enabled our ancestors to have as good a sense of smell as dogs. But the genes have mostly been turned off. And so we have vestiges, we have historical relics of those genes. And it's like your hard disk on your computer that's cluttered up with remains of old chapters you've written here and there and things that have now been cut off. Those genes have been turned off, but they're still there.
Isn't that the premise of X-Men?
I don't know.
They're human, but they have a genetically different genes are turned on and off within them, giving them special powers.
Language is a window into social relations. I'll begin with a puzzle in language.
This one is taken from the movie Fargo, from a scene early in the movie in which a kidnapper has a hostage tied up in the back seat of a car and inconveniently is pulled over by the police because he's missing his plates. The police officer asks him to show his driver's license. He proffers his wallet with the license showing and a $50 bill extending ever so slightly. And he says to the officer, I was thinking that maybe the best thing would be to take care of it here in Brainerd, which the audience and presumably the officer recognize as a veiled bribe. Now, this is an example of what linguists call an indirect speech act, a case in which we don't blurt out what we mean in so many words, but we veil our intentions in innuendo, hoping for our listener to read between the lines and infer our real intent. And this is something that we do all the time, often without realizing it. For example, if you could pass the guacamole, that would be awesome. Now, when you think about it, that doesn't make a whole lot of sense. But we effortlessly recognize it as a polite request. We're counting on you to show leadership in our campaign for the future. Anyone who has sat through a fundraising dinner is familiar with euphemistic schnorring like that, which can be translated as, give us money. Would you like to come up and see my etchings? That has been recognized as a sexual come-on for so long that in the 1930s, James Thurber drew a New Yorker cartoon in which a man says to his date, you wait here and I'll bring the etchings down. Then there's a nice story you got there. It would be a real shame if something happened to it, which any viewer of The Sopranos could recognize as a veiled threat. So the puzzle is, why are bribes, requests, seductions, solicitations, and threats so often veiled when both parties presumably know exactly what they mean? Language has to do two things. It's got to convey some content, such as a bribe, a command, or a proposition. At the same time, it's got to negotiate a relationship type. The solution is to use language at two levels. The speaker uses the literal form to signal the safest relationship to the listener, while counting on the listener to read between the lines to entertain a proposition that might be incompatible with that relationship. And politeness is a simple example. What's going on with, if you could pass the guacamole, that would be awesome? I think everyone would agree that it's a bit of an overstatement. And also, it's not clear why you should be pondering counterfactual worlds right there and then at the dinner table. Now, the listener thinks, assuming that the speaker has not lost his mind, the speaker says an outcome is good, therefore he must be requesting it. The overall effect is that the intended content gets through, namely the imperative, but without the presumption of dominance that would ordinarily accompany an imperative, namely an expectation that you can be commanding some other person to do what you want. Well, according to the anthropologist Alan Fiske, there are only 3 major human relationship types across the world's cultures. Each prescribes a distinct way of distributing resources, each has a distinct evolutionary basis, and each applies most naturally to certain people, but can be extended through negotiation to others, and that's where language comes in. So there's dominance, as I've mentioned, whose logic is don't mess with me, and which presumably we inherited from the dominance hierarchies that are ubiquitous among primates. Very different from that is communality, the ethos share and share alike, which evolved by a different route, namely kin selection and mutualism, and therefore is extended by default to kin, to spouses, and among close friends. Finally, there's reciprocity. You scratch my back, I'll scratch yours, which pertains to the business-like tit-for-tat exchanges of goods and services that characterizes reciprocal altruism. Now, behavior that's acceptable in one relationship type can be anomalous in another. For example, at a drinks party, you might go over to your husband or wife or boyfriend or girlfriend and help yourself to a prawn off their plate. But you wouldn't go up to your boss and help yourself to a prawn off his plate, because what you can get away with in a communality relationship, you can't get away with in a dominance relationship. Likewise, at the end of a dinner party, if you pulled out your wallet and offered to pay the host for the dinner, that would not be perceived as fair, that would be perceived as crass, because of the clash between reciprocity, which is what would be appropriate, say, at a restaurant, and communality, which is what we deem appropriate among friends. Now, those are cases where everyone knows what's appropriate, but in cases where the two sides aren't sure that they're on the same wavelength, a divergent understanding can lead to an unpleasant emotion, the one that we call awkwardness. For example, there can be awkward moments in a workplace when an employee doesn't know, or a student doesn't know whether to address a supervisor by their first name or to invite them out after work for a beer because of the ambiguity as to whether their relationship is governed by dominance or friendship. It's well-known bit of wisdom that good friends should not engage in a major business transaction, like one of them selling his car to the other. The very act of negotiating a price can put a strain on the friendship because what's appropriate in a reciprocity relationship is not appropriate in a communality relationship. The contrast between dominance and sex, as when a supervisor solicits sex from an employee, defines the battleground of sexual harassment. And even the two kinds of communal relationship of friendship and sex give rise to the anxieties of dating. Well, one remaining problem, which is why we resort to indirectness even when there is no real uncertainty. For example, when the listener knows the speaker's intent. People aren't naive, and it's hard to believe that any grown woman could be fooled by the line about the etchings. Nonetheless, there is something that is more comfortable about asking to see etchings than asking for sex. So what is going on there? The deniability is not really plausible. Why should an obvious innuendo still feel more comfortable than a direct overture that is in some sense on the record? To illustrate the problem with a scene from the romantic comedy When Harry Met Sally, where in an early scene of the movie, Harry makes a remark that Sally interprets as sexual, and she accuses him of, "You're coming on to me." So he says, "Well, what do you want me to do about it?" "I take it back, okay? I take it back." She says, "You can't take it back." "Why not?" "Because it's already out there." He says, "Oh, geez, what are we supposed to do? Call the cops? It's already out there." Well, what is the psychological status of an overture that we feel to be out there or on the record that makes it feel so much more awkward than a veiled overture that's conveyed indirectly? And I think a key to this paradox is a concept that economists and logicians call mutual knowledge, which they distinguish from individual knowledge. In individual knowledge, A knows X and B knows X. In mutual knowledge, A knows X, B knows X, A knows that B knows X, B knows that A knows X, A knows that B knows that A knows X, ad infinitum. And this is a difference that has profound consequences. For example, why is freedom of assembly enshrined as a fundamental right in a democracy? And why are political revolutions often triggered when a crowd gathers in a public square to challenge the president in his palace? Well, it's because when people were at home, everyone knew that they loathed the dictator, but no one knew that other people knew that they knew. Once you assemble in a place where everyone can see everyone else, everyone knows that everyone else knows that everyone else knows that the dictator is loathed, and that gives them the collective power to challenge the authority of the dictator, who otherwise could pick off dissenters one at a time. Another example is that The Emperor's New Clothes is a story about mutual knowledge. When the little boy said the emperor is naked, he wasn't telling anyone anything that they didn't already know, anything that they couldn't see with their own eyeballs. He was nonetheless changing the state of their knowledge, because at that moment, everyone now knew that everyone else knew that everyone else knew. Once again, that gave them the collective power to challenge the dominance of the emperor through their laughter.
What we found was that kids with autism produce pretty much only one kind of laugh, whereas typically developing kids produce different kinds. And the main difference is what we call voicing. So in a prototype laugh, like the one we'd think, just when you think of a laugh, what comes to your mind is that laugh that we produce, we're using our vocal cords to produce it. Just like I'm talking right now and I'm using my vocal cords. Another kind of laugh that we do in adults, about half of the laughs that we do are called unvoiced laughs. So this would be things like a snort, like a, or a shh, stuff like that. So I'm not using my vocal cords to produce it. As it turns out, the kids with autism produce 98% of voice laughter, at least between 8 to 10. Whereas typically developing kids produce about 40% or so in that same age range. And so We have data showing that people have a strong preference for listening to voice laughs as in comparison with unvoiced laughs. Like in our conversation right now, if I say something dumb, you might kind of, thank you, exactly. And you don't really feel good inside, but you've learned to sort of grease the social wheels in this context. Whereas with kids with autism, I don't think that they're probably picking up on those subtle social cues. So it's probably more, the kinds of laughs that they're producing are probably more heartfelt and that they're genuine. So they just laugh when they feel good. Kids are producing these sounds that people really like to hear and that may actually influence others positively.
The moral of the story is that explicit language is an excellent way of creating mutual knowledge. And so here's the hypothesis. Innuendos, even obvious ones, merely provide individual knowledge, whereas direct speech provides mutual knowledge, and relationships are maintained or nullified by a mutual knowledge of the relationship type. So if Harry were to say, would you like to come up and see my etchings? And Sally says no. Then Sally knows that she's turned down an overture, and Harry knows that she's turned down a sexual overture. But does Sally know that Harry knows? She could be thinking, maybe Harry thinks I'm naive. And does Harry know that Sally knows that he knows? He could be wondering, maybe Sally thinks I'm dense. There's no mutual knowledge, and they can maintain the fiction of friendship. Whereas if Harry were to say, would you like to come up and have sex? And Sally turns him down. Now Harry knows that Sally knows that Harry knows that Sally knows. They cannot maintain the fiction of a friendship. And I think this is the basis for our intuition that with overt language, you can't take it back. It's out there.
Let's get back to our interview with Dan Aykroyd. I read that when you were 12, you were diagnosed as schizophrenic, and that you had to kind of keep that under control.
Well, it was more of a Tourette's thing than schizophrenia. It was analyzed as a Tourette's and Asperger's, which I still have a little bit today.
Hello, everybody, and welcome back to New Books in History. I'm Marshall Poe, your host. This week we're happy to have Gregory Cochran on the show. I guess I just want to press you here just a little bit because I find this very interesting. What were the specific traits that you think were selected for?
I think that people were selected for submission to authority once there was such a thing as greater authority. Because I think that if you don't, in many situations, you ended up dead. So I think that in hunter-gatherers, and this is a lot of this is from saying what Henry Harpending spent quite a long time with the Bushmen, but they don't have bosses. Yeah. They don't really have tribal chieftains. They do have some people that people say, yeah, we should probably listen to that guy. He's pretty sensible, but that's about it.
Yeah.
And when somebody tries to be a boss, they make merciless fun of you.
Yeah.
Of course, in primary school and high school, I imitated all the teachers, and then I met guys who could imitate the teachers better than me, and then we sort of formed, these imitation squads, and, my grade 11 math teacher, Father Paul Baxter, God rest his soul, I was not one of his favorite students. There were 10 of us that did him, and we would have, one time we all came dressed as him to school in the white lab coat with the little Coke bottle glasses, and he kind of had a backward walk. He kind of wheeled back to the blackboard. And he had this voice like it was just, okay, jokers, smarten up.
Agricultural peoples have bosses. They have bosses of bosses. They have hierarchical civilizations. You're under a lot of other people's influence and control. There's all sorts of people telling you can't do this and you have to do that. And I think that it's easier for some people, certainly today, it's easier for some people to put up with this than others or to fit into it than others.
And at 12 years old, was able to have the luxury of sitting down with a therapist and talking through all kinds of things, books, and music, and she was quite influential in kind of evening me out.
If there's someone here who has an autistic child or knows an autistic child and feels kind of cut off from them, what advice would you give them?
The thing is, autism comes in different degrees. There's going to be about half the people on the spectrum that are not going to learn to talk, and they're not going to be working in Silicon Valley. That would not be a reasonable thing for them to do. But then you get these smart, geeky kids, the touch of autism, and that's where you've got to get them turned on with doing interesting things. I got social interaction through shared interests. I rode horses with other kids. I made model rockets with other kids.
And I just find in my research and reading today that there's a lot of people who have this kind of mild condition, and Some of them get over it, and some of them it spins out where it affects them quite negatively. Well, me, I have a fascination with law enforcement and the police. I guess my grandfather was a Mountie and that, so if I don't have a badge on me, I feel naked.
Well, I can't tell if you're kidding or not. I know you studied criminology when you were in college.
I did. I studied criminology. I had a great professor there, and that study of criminology really helped me when I wrote the Blues Brothers, because the Blues Brothers were classic recidivists, could never stay out of trouble.
It's important to discuss the goals of our society with respect to neurodiversity, which is one of the things we might learn from a detailed understanding of Neanderthal. They're almost certainly different from us in the way they think, and also it could be illuminating about how we approach the issues of biomedical safety and discrimination and stigmatization and so forth.
I'm going to finish just by talking a little bit about empathy outside of the human species, because having identified that genes play a role in empathy, it raises this idea that empathy may be the result of evolution, and that those genes may have been selected, positively selected, during evolution. The only experiment that I've come across which has tried to systematically test empathy in monkeys was by Jules Masserman at Northwestern University in the 1960s. And what Masserman did was train these rhesus monkeys to pull a chain every time they wanted food. And the monkeys soon learnt that if they did that, they got the reward. And then Masserman changed the contingencies so that when they pulled the chain, yes, they got the reward, but they also saw another monkey getting an electric shock. And what Masserman observed was that the monkeys soon stopped pulling the chain as soon as they learnt that in getting a reward for themselves, it was also causing pain to another member of their species. So this certainly suggests that simpler forms of empathy may predate human beings, which should come as no surprise.
Is it unrealistic for them to hope or think that that child loves them, as some might most wish?
Well, let me tell you, that child will be loyal, and if your house is burning down, they're going to get you out of it.
It's very loaded.
Yes.
You know what I mean?
The thing, though, is that I have the feeling that how we speculate about the Neanderthals, I often like to say it says more about our worldview than anything about what happened back then. I don't think that there is any sort of scientific knowledge or insight that will actually convince people to really change their ingrained ideas about this. One is often asked, why do you think Neanderthals disappeared? Did we kill them all? If you like to see modern humans as very violent, you will say, this was obviously our first big genocide, just look at how we behave today. On the other hand, if I say the earliest modern human fossils in the Middle East are 93,000 years old, the latest Neanderthals in the Middle East are 60,000 years old, so we have 30,000 years of peaceful coexistence, and that in the Middle East. If we could have that today, it would be wonderful. Now you can say, well, that's probably because modern humans came, then Neanderthals disappeared, and modern humans disappeared again, so they never really interacted with each other. But who knows? It just reflects how we think of ourselves in a way, what stories we make up of what happened back there.